Hello everyone, I'm Mr. Furlong, and today we're talking about Reginald Punnett. I'm sure you never heard of Reginald Punnett before. He came up with a simple way to show how traits can be passed from parent to offspring. So let's take a look at how he did this. First of all, Mendel said that all factors or genes, or what I'll be calling alleles, can either be dominant, which means that they're going to be expressed, or they're recessive, which means that they may not be expressed. Their trait may be masked by a dominant allele. And in a Punnett square, we use letters to represent these alleles. A dominant allele is always represented by a capital letter, and a recessive allele by a lowercase letter. What letter you choose is totally up to you. There's no set rules on this. However, I would stay away from letters in which the uppercase and the lowercase look similar. So for instance, O's and S's not as good as say A's and B's. A few definitions that you're going to be seeing quite a bit of. Homozygous means that both of the alleles are alike. They're the same. So for instance, an individual could have two dominant alleles for a trait, or they could have two recessive alleles. We would say that they are homozygous dominant or homozygous recessive. If the alleles are different from one another, we would say that that individual is heterozygous. In other words, they have one dominant allele and one recessive allele. We also need to identify what a phenotype is. The phenotype is the observable trait, or in other words, it's how that trait is expressed. So let's look at this idea of eye color. Brown eyes are dominant to blue eyes. If a person has brown eyes, we can see that. So the phenotype is observable. I can tell just by looking at the organism what their phenotype is. But I don't necessarily know what their genotype is. For instance, this person with brown eyes could be homozygous dominant, or they could be heterozygous. Both would show the same phenotype. Right, so that is the genotype. Well, what is the genetic makeup of that person? Or what's the combination of genes that they have that give them that trait? For instance, they might be homozygous dominant for brown eyes. Let's take a look at how we would use a Punnett square. In one of Mendel's early crosses, he took a homozygous dominant tall pea plant and he crossed it with a homozygous recessive short plant. And we are being asked to find out what are the phenotypes and the genotypes of the offspring. To do that, we are going to make a Punnett square. Now a Punnett square is simply you make a big box big enough to write some letters in, and then we're going to divide that into 4 smaller boxes. We have to identify what are the genotypes of the parents. So we have some clues. For instance, we have that one parent is homozygous dominant tall. Now I'm going to use a few letters here. I'm going to use a capital T to represent the tall allele and a lowercase t to represent the short allele. So I have one parent who is homozygous dominant for tall and is crossing with a homozygous recessive short plant. So that would be 2 little t's. I put an X with a circle around there just to represent this as a cross. Now I'm going to take one of the parents and I'm going to separate those 2 alleles because only one of them will actually be given to the offspring, and it's by chance as to which one it is. So I take one T and I'm going to put it above one column, and I'm going to take the other T and put it above the other column. Then I'm going to take the other parent and I'm going to separate their 2 alleles. Put one in front of each row. Now could I have done this the other way? Certainly. There's no rules on which parent goes on which side of the Punnett square. Now in the box I'm going to show the combination of alleles. So this is going to show me 4 possible outcomes from this cross. So I will bring the dominant T down and the lowercase t over. And now I have one possible outcome. I'm going to do the same thing in this box. And down below. Let's start with the genotypes. All of these are the same. So the genotypes are all big T, little t, or heterozygous. The phenotypes, because the dominant allele says tall, the recessive says short, because the dominant alleles are always expressed, this is going to be a tall plant, tall plant, tall plant, tall plant. So all of these offspring will be tall. When Mendel did this, thousands of plants were tall. So then he took two of these tall plants and crossed those together. Now these two tall plants is from the first generation, or the first filial generation, the F1. And now he was crossing them to find out what their phenotypic and genotypic ratio is. So let's do this cross. First we need to know what the parents are. Remember, all of the offspring in that first generation were heterozygous. They were big T, little t. So we're going to take one parent, separate the two alleles, put one in front of each column, and then we take the other parent, separate the two alleles, put one in front of each row, and now we're going to show all the different possible combinations. So there is a offspring that is homozygous dominant. There's a heterozygous one. There's a heterozygous one. And there's a homozygous recessive one. Now this is asking us not just for the phenotype and the genotype, but it wants to know what is the ratio. So let's start with the genotypic ratio. We have one homozygous dominant, and that's usually what we start with, are the dominant ones. We have two heterozygous, and one homozygous recessive. So we would say that the genotypic ratio is 1 to 2 to 1. The phenotypic ratio, in this case, we have two phenotypes, tall and short. So this was a tall plant. That's a tall plant. That's a tall plant. And that's a short plant. So we had 3 talls to 1 short, the phenotypic ratio. And so when Mendel grew those plants, this is exactly what he saw. 3 talls for every 1 short. Or another way of writing this is to say that of the 4 possibilities, there's a 75% chance of having a tall plant, 25% chance of having a short plant. And in fact, when Mendel grew thousands of these plants, about 75% of them came out tall, 25% came up short. And that's all it is. So we're going to be practicing a bunch of these in class. We're also, in the next couple of videos, going to be looking at some more complex types of Punnett squares. And I can't wait to tell you all about it.